Please join me in welcoming Dr. Christopher Hendon. Thanks, Chris.
Thanks, Jennifer. Thank you very much for the invitation. And thank you, everybody, for taking time away from your this evening to tune in and listen to this talk on the Distinctive Voices talk hosted by the National Academy. So it's my pleasure to talk to you today about the chemistry and physics of producing coffee. This is a topic that I'm certainly not employed at the University of Oregon primarily to study, but I've been working in it, and it's a topic that we've recently got funding to continue to pursue. So hopefully there'll be more of what you're going to hear tonight. You heard a little bit about my bio when we started just then, but actually, as it turns out, that I have my DNA sequenced, and the oldest known ancestor of my bloodline is 60,000 years ago, roughly. And you can see that pin is around here, which incidentally is very close to Ethiopia, Kenya, which is basically one of the birthplaces of coffee. So one might argue I've been actually involved in coffee for quite some time. As Jennifer mentioned, I did my PhD at the University of Bath, which itself is a relatively old city. If you've ever visited, all of the buildings look beautiful. They look like this. And it's named Bath because the Romans settled here and basically constructed these baths around a hot spring. You can still go today to the baths and walk around. Of course, you shouldn't be bathing in them, but it's a wonderful thing to do if you ever get the opportunity. And shortly after I finished my PhD, I spent 2 years as a postdoc between the Department of Chemistry and the Department of Chemical Engineering at MIT. And it was there where I sort of refined my conceptual understanding of how to put chemical processes into larger scales, which is basically what coffee is. And then really started to develop myself as a scientist. And then shortly after that, I went on the job market and I got a tenure track position at the University of Oregon. Now, you may know Oregon because we had a pretty good football team. We're not bad right now as well. This is Autzen Stadium. Of course, if you were to see a game on TV, there are no fans at the moment. But we're also famous for a few other things. We've had some academic success. We're also the home of Steve Prefontaine. And of course, there's also Phil Knight, who's the founder and ex-chairman of Nike. At the University of Oregon, I lead a small research group, which is primarily a computational chemistry group. And it is actually these students that really do all of the hard work, and really all of our success comes from these 8 talented individuals. On the top, we have graduate students, and on the bottom, you can see I have 4 very talented undergraduates, one of which is actually going out in the grad school pool this year. So keep an eye out for Jack Yang's application. And we study topics ranging from metal organic frameworks, which are porous materials, all the way to really fundamental properties of molecules. For example, their chiral response, their optical response to light. Macroscopically, you can define what we do in the group as basically being interested in performing chemistry with earth-abundant elements. And I define abundant as anything above this line that I've drawn here, which is one atom per million atoms of silicon available in Earth's crust. And so I point to a couple of examples of catalytically relevant industrial metals like platinum, iridium, and rhodium. And actually, these are very useful, but they're also very expensive, and there's other problems with them. And so my group is interested in basically obtaining the same style of reactivity and same type of reactivity you might achieve with platinum, iridium, or rhodium, but using earth-abundant metals. Now, nature can do this, right? Nature does this with enzymes, and they use essentially multinuclear clusters and enzymes to perform similar types of catalysis. With relation to metal organic frameworks, which is what my group's primary discipline is, we have these site-isolated, basically small regions of inorganic chemistry that are sort of suspended in 3D space. And in this particular example, this is a nickel center that is supported by some organic linkers. And this nickel center is capable of performing a dimerization, the coupling of 2 ethylene molecules together to form selectively 1 butene. Now, this is a valuable transformation because 1 butene is a valuable chemical feedstock for the polymer industry. So to be able to catalyze this using an earth-abundant metal is of course something that we'd be interested in. And it's something my group actively works on. But that's not what we're here for today. In fact, we're here for something seemingly orthogonal. And I apologize that we may not have time to cover everything that you might want to talk about. So first of all, I'd encourage you to use the Q&A feature in Zoom. Second of all, if there's something that we don't get to and it's not covered in any of these talks that you can find online, please send me an email. I love having these discussions. But otherwise, hopefully these talks that I've also had recorded will perhaps entertain or answer some of your questions. Additionally, I curate a repository of some papers that I find interesting and somewhat relevant to coffee science in general. Of course, these are not formally hosted on UO WebSpace, for legal reasons. I say that they actually are, of course. But the point is that these are here so that people in the coffee industry can read peer-reviewed articles that may relate to things that are important to them. For example, this 1979 paper that talks about the misuse of the word sour when describing bitterness and vice versa, and so forth. So if there's a paper that you've come across, or maybe one of your papers on the topic that I don't have hosted there, please send it to me, I'll host it. My coffee journey begins by walking into a cafe, and I'm greeted with the board that looks like this. This is the menu. And what you can see here is on the left are drinks that are made as an espresso, and on the right are filter coffees, low concentration coffees. And then on the left, you can also see things like the country, the processing methods such as natural or washed, and you can see varieties, much the same as you might peruse a wine list. And then associated with these things is also a flavor note. So if I were to have an espresso of this particular Brazilian coffee, I might, as a consumer, expect to taste the notes of fruitcake, brandy, and toasted seeds. But interestingly, if I add milk to it, then I might have that same espresso no longer taste like fruitcake, brandy, and toasted seeds, and now tastes like dark toffee, custard, and Brazil nuts. And then interestingly, if I compare this panel to perhaps the one below it from Rwanda, I do see some similarities. I see treacle, of course, which is sort of like a gooey sweetness. I see spices, I see candied orange, and I also see port and brandy, so I see sort of a boozy note. But I don't really know where the flavors are coming from. Are these flavors, fruitcake, brandy, and toasted seeds, are they unique to Brazil? Or is it unique to the natural process or the variety, or is it some combination of all of them? And this is the sparse matrix that I was given as a standard consumer when I was a graduate student. But zooming out, the aim in the coffee industry, at least at the point of consumption, is not for me to try and tell you what the best flavors are, nor am I trying ever to sterilize or standardize your taste. In fact, I embrace what you choose to enjoy. In fact, I'm viewing it from the other side. But where if a cafe told you that they wanted the coffee to taste like lemon peel, bergamot, and watermelon, then indeed you would be hoping to taste those flavors. And if you were then to order that coffee and achieve salmiakki dirt and Brussels sprouts, perhaps there is a language disparity, but perhaps there's something chemically wrong in this process. And so at this point, we can ask, well, what could have given rise to the total complete change in flavor profile from nice, lightweight, fruity acids to tasting somewhat like dirt? And if we zoom out again, we could say, okay, 50% of that variation in that cup could come from the quality of the natural product, quality of the green coffee. And then 20% from the roast, 20% from the water chemistry, and 10% from the equipment. But you can't isolate just those pieces because the coffee supply chain is complicated. And I've simplified it here, extremely simplified it here, where basically you have a producer, a farmer. They may or may not then process the coffee themselves. At some point, it has to leave the origin, and then it has to be imported into the country where, for example, the United States, where I am, and I'm going to consume it. It then needs to be roasted, and then it needs to be distributed. And so along the way, there's time as well. And time, of course, for natural products it gives rise to changing properties. And I also point out that equipment is only about 10%, because, of course, we can romanticize about equipment. We could spend any amount of money possible on equipment to prove a point. Here's a cup that NASA developed so that people can consume coffee in zero gravity. Basically, the water, it's curiously shaped and colored, but the liquid goes inside the hole here, and then through wetting, the liquid pools along this sort of capillary, if you like, along the edges, pooling at the lips here. You put this in your mouth and you drink from it. Of course, this is probably the most expensive example of a coffee cup, but perhaps the least expensive example, but the most relevant to the industry, are these 5 small cups on this table. And basically, all coffee is graded on a cupping table. And all this is just grinding coffee, pouring hot water on it, waiting a few minutes, and then tasting it. And you're tasting 5 cups or more than 1 cup of the same coffee to ensure, that there's no variation within the same crop. So this is why I'm saying the 50% comes from the green. And you're also then just looking for the flavors that this cup exhibits over time, okay? Now, to assess the qualities of coffee, there is a way of standardizing tasters. And, this isn't necessarily for me as a scientist who studies this, or for you, perhaps as a scientist who may study it, or a consumer. This is more of an expert or a professional's approach to tasting coffee, where they have a score sheet, and the highest score on this sheet would be a cumulative score of 100. The lowest score would be a cumulative score of, well, actually, I don't know. I've never had an extremely low scoring coffee, so I don't really know. But the important point here is they're subdivided into panels, where some importance is given to fragrance and aroma, flavor, aftertaste, acidity, and so forth. Okay, but I've blown up one panel in particular, acidity, because this panel is extremely important to the way that we perceive coffee. Now, if you look at peer-reviewed articles that have been published related to acidity and sweetness in model systems like liquids containing citric acid and fructose, there seems to be a report in these papers that say that as acidity level increases, perceived sweetness level decreases. And so this is tricky because sweetness is over here as a panel and acidity is a separate panel. So scoring high on this is very difficult. You basically need to have all of these things, all these boxes ticked. But I simply want to plant in your mind that acidity, the panel acidity, is extremely important to getting to the final high score. Now, any coffee that scores above 80 points is deemed a specialty grade coffee, and these coffees fetch a much higher value per pound for the farmer. So there is an incentive to try and score high on this score sheet. At the same time, I also want to point out that if you don't like acidic coffee, that does not mean that you're a Philistine. In fact, it probably means you're going to save some money, because the coffee itself may score low in perceived acidity, therefore scores low overall on the score sheet, therefore costs less money. Okay, but we'll get into that as we progress through. To start the talk, we need to go back to the plant and to think about the economics. So in the United States in 2015, the coffee industry is worth 1.3% of the US GDP. This includes import, export, it includes cups, straws, jobs, retail, everything, okay? And within that, if you think about how coffee is consumed in America, we primarily drink drip coffee, which is 1.2 to 1.5% by mass coffee solids solvated in water. In Europe and in other coffee consuming cultures, higher concentration coffee such as espresso is typically favored. And that is in the vicinity of, let's say, 8 to 10% by mass solvated coffee. So let's call it 10% as 10 times concentrated, right? Now, this is a pretty important to our economy, but it's really important to places like Burundi, where Burundi's 25% of their GDP comes from the coffee industry, but 90% of the population is employed in the coffee industry. And so a bad year for buying coffee from Burundi is a bad year for basically everybody in Burundi. Burundi, of course, is just one little tiny country in a variety of other countries that produce coffee. This sometimes is referred to as the bean belt, but in essence, what you want to produce coffee is relatively stable, warm temperatures, and you want relatively low carbon dioxide concentration. Because remember, what you're trying to target is the seed, not the fruit that surrounds it. And so if you have high CO2, the fruit will mature much the same way that a seedless grape matures. You get a lot of fruit and then not much seed, right? Now, within this belt, there's Robusta and Arabica are two different species that are grown. And so they're actually very dissimilar. Arabica has twice the number of chromosomes, which is not a good indication, of course, of flavor complexity or even of biological complexity, but simply noting that these are really dissimilar plants, right? But importantly, the Arabica actually is susceptible to pests because it produces far less caffeine per gram of coffee material, which means that caffeine is the plant's way of protecting itself from little bugs. And so a Robusta can grow at lower altitude where there's a lot of bugs, and Arabica at higher altitude. I also want to point out that coffee plants are, I wouldn't say easy to grow, but we can grow them inside here in the United States. This is my coffee plant, Jean-Paul, who lives in my kitchen. He's Arabica, and he's a very, very long way away from fruiting. But if he were to fruit, like the other plants that produce commercial coffee, they produce something that looks vaguely like a cranberry. And if you were to split this thing in half, this coffee cherry in half, you would typically find two seeds. If you find one, that's called a pea berry defect. And indeed, it's not really a defect. It's embraced in Kenya now as a Kenyan PB. But let's assume you've got these two seeds. You then can remove all the fruity mucilage and skin, and inevitably you'll get down to what is actually the seed. Now, this is sort of the if nature had its way with the plant would fruit, it would then ripen, maybe fall off the tree, and then in the sun it would shrivel up to a raisin type material, and then inevitably maybe it's eaten, maybe not, but the skin and the raisin are surrounding the seed. So we can intervene and we can basically perform this process ourselves. This is called the natural process. We pick the coffee off the plant. We then dry it out in the sun on a raised bed, and it turns into something that looks like a craisin. Then we somehow remove the skin and the sort of fruity stuff that's remaining, and we're left with the seed. That seed then can be fermented. Other things can happen, but inevitably you get something that looks like this, but it's gone through a different process. The wash process is where you use water to remove all of the skin and the fruit. And so you just get to something that looks like the seed pretty rapidly. And then we have the pulp process, which I have an example of, where you remove some of the skin, but you leave some of the fruit behind, and then you dry that in the sun. And so it looks a bit like this. And then along the whole processing method, chemicals are developed, right? And I use the word chemicals loosely here because there is definitely different flavors that come from the natural process. And we haven't even roasted the coffee yet. But identifying those as precursors is something that's an ongoing research project for many people. At some point, then the coffee is fermented, and it can happen while still in the skins. It can also happen in this stage as well. Now, naturally processed coffees typically come from places that had struggled with water. And so that would be places like Yemen, Ethiopia, et cetera. So here is a picture of a Yirgacheffe coffee production facility, and you can see these are a naturally processed coffee. These are cherries that have been picked. They may be being sorted at the moment, but the point is, they're going to sit on these raised beds and dry out. And then in the background, you can see what may or may not be washed coffee drying in the sun as well. Now, what we do know is that coffees that have been naturally processed occasionally have issues with bacteria, fungus, and other things growing on them. And those defective beans tend to fluoresce under a black light. The chromophore, or fluorophore that results in this fluorescence is not well identified. That is something that we probably should look into. So at this point, we've got some coffee, it's green, okay? And I want to point out that once it's green and sitting there, it's still active. So the coffee itself is picked with about 12.5 to 13.5% water contained within the seed, but it's going to be in an atmosphere that is lower water than that. And so it's going to be losing water through water activity. And so by the time you actually roast it, it actually has less water in it. But the water at the roasting stage is critical because water has very high heat capacity, so it changes the way that you have to think about roasting. We actually have a project ongoing at the moment with one of my graduate students examining and trying to use machine learning to predict the degradation and properties of coffee based on myriad variables that give rise to differences in quality over time. I think this is an excellent project for machine learning. So at this point, you're probably thinking, well, what's the best? Because I've shown you natural and washed and pulp natural and so forth. And typically, you've probably heard of the Kopi Luwak or the civet coffee. Well, this is basically a poo. And so what's happened is a small animal has eaten coffee cherries. They've used the fruit for fuel for their body. The seeds make it through their digestive tract, and out they come like this. And so basically, that's basically the washed process, right? It's been fermented. This is definitely not the best coffee in the world. This does not score very high on the score sheet, not that I've ever seen. And the best coffee in the world tends to not have a particular country dependence. It changes year to year based on the highest scoring coffee in a variety of other competitions and so forth. But I can tell you with certainty, this coffees that score low on this score sheet are ones that have low perceived acidity. And those are from Brazil, Sumatra, Vietnam, and Hawaii. And then coffees that typically score high on this score sheet are this variety called a geisha, either a natural or a washed. Now, I want to also point out that these geishas are sometimes referred to as geisha, sometimes used interchangeably. We can have a discussion about that afterwards if you'd like. But the important point is this has no relationship with the Japanese hostess whatsoever, instead related to what is thought to be a producing region in Ethiopia. And so again, the point is the best coffee in the world really is one that scores highest on these score sheets. So at this point, you've got a green bean. And fundamentally, you now need to turn it brown. So you just have to roast it. And if you were to zoom out and think about what that looks like, well, you start out at room temperature, and you got to heat it up. And at some point, you're going to hear an audible crack. That crack is associated with water vapor leaving the bean, carbon dioxide that has been generated from decarboxylation reactions leaving this bean, and so forth. And then you're going to continue roasting perhaps, and you'll then eventually hear an audible second crack. And then if you keep roasting, you're going to get to something that the beans start to sweat, and you can keep going, and eventually they'll catch on fire, right? but it's not really about the color. It's actually about how you got there. And so the roast profile is really a kinetics problem, because basically what you do is you start with a heated up cement mixer, which is the roaster. And here, as I've shown you an example of one, this is from the Starbucks Reserve in Seattle, Washington. This is a really big hot chamber. And then you drop in room temperature coffee. And when you do that, the heat transfers to the bean. That's called the soak. Then as you come out of that little gradient there that you come out of really actually plays a critical role in determining the roast that you're going to achieve. And so then what you get as you come out of the soak is you have to then control the gradient of the increase of that temperature there as you generate different products that you may or may not like in the roast. Now, typically, people tend to think that a nice smooth rate of rise, a declining rate of rise is a good thing and gives rise to good flavors. Here you can see this is a crash rate of rise, which means that at the end the temperature, the gradient changed rapidly, perhaps not ideal. Okay, but the point is the roaster is controlling this with two knobs. They're controlling it with airflow and they're controlling it with fuel, much the same as running lean or rich. Okay, and so it's very difficult to do this reproducibly, and it's done largely manually. If you give it to a scientist, we'll do something totally orthogonal. So we'll take the coffee, we'll brew it, and in a paper in ACS Applied Materials and Interfaces in 2017, the authors went on to then expose that same coffee to zinc chloride under an argon atmosphere at 800 degrees Celsius with the aim of making N-doped carbon. And when you do this, this material actually is pretty good at forming oxygen reduction catalysis, much the same as what we use platinum for. So basically you take O2 with a source of protons around and you make water. This is important because this is like one of the reactions that has to happen in a fuel cell. So, potentially you could maybe use roasted coffee that's been re-roasted for this. I don't know. This is a bit of a stretch. What's actually in roasted coffee is more important. Well, it turns out there's a fair bit. That's a quarter of the compounds that were found in the headspace. And so the point is that roasting coffee, again, while you can apply chemistry to this, it's still an art in the sense that controlling the relative ratios of these things depends on the roast. One thing that may or may not depend on the roast, however, is the caffeine content. In a paper in Science in 2014, they isolated sort of in the chain of biology where caffeine is produced and its similarity in coffee to other plants that also produce caffeine, like tomato, sweet potato, and so forth. But interestingly, caffeine itself, if it were a pure crystal, melts at 235, but boils at 178. In other words, it sublimes, okay? And so we've observed this too. I was trying to take a surface area measurement back when I was at MIT, and the caffeine sublimed and then recrystallized up here in this tube. So that was a real pain in the neck. But the point is this, is that as you start to roast coffee, the coffee begins with a finite amount of caffeine. And the more you roast, the more you're going to sublime the caffeine away. But at the same time, the more you roast, the more accessible the caffeine becomes because you've roasted the coffee. And so it turns out that a dark roast and a light roast don't really have that different level of a caffeine. And I could point you to some papers, but people have stopped worrying about caffeine because, as I'll remind you back, if you were a little critter, caffeine is in fact a compound that we don't really like, nor do the bugs. It tastes very bitter. And so we much prefer to think about other flavors. I also want to point out that there are free radicals in coffee. This is pretty standard. If you perform a Maillard reaction, you almost always get free radicals that are forming from small little carbon flakes that show up in places, and much the same as you'd find in beer. So instead of thinking about chemistry, what the coffee industry has done is they've said, why don't we standardize words that we would use to describe flavors? So going around the wheel on the outside are flavors that a trained set of tasters have tasted in a multitude of coffees. Now, not a single coffee I've ever encountered has all of these flavor notes in it. It typically only has maybe 5 or 6, perhaps, okay? But the point is this, is that for every word on the outside of this ring, there's a standard way of preparing that flavor. So that independent of whether you and I agree that that standard tastes the way as that word says and the way you associate it with your experiences in your life, we can reprogram ourselves to now identify peach as peach with reference to the standard state that they've given us, okay? And so, for example, for peach, the reference for peach is peach jello but without the water added, okay? Okay, so with that in mind, you can go all the way around this ring and you can look up the lexicon here at World Coffee Research. And this has been useful because it teaches us how to use the same language. But we don't really know what's in the coffee yet. We've got a green bean that's been roasted. So now we need to grind it up. And now fundamentally grinding is very difficult to talk about because if I told you to grind it coarse or medium or fine, without having some sort of measurement for that, it's almost impossible. So very loosely, I've written coarse here, but I mean, who knows what that means. So what we actually do is we take ground coffee, whether we use a whirly blade grinder, you probably got one of those in the kitchen, a conical burr grinder or a flat burr grinder, these are two standards in the industry. And we take the particulates and pass them through a device which uses diffraction of light to measure their size. In short, what happens is this little hollow point drill bit does not actually regrind, but separates the particles, passes them over this light source, and we're able to then make an assessment of the size of coffee. But the problem with this is that, of course, the coffee may not be a perfect sphere. And so the question is the same as how big is the sea urchin? Because is it this big, or this big? Well, of course, the sea urchin is this big because that is exactly the size that it takes up. But the diffraction method actually gives you some average size. And this is probably a little generous. It's probably a little smaller than this, but it's going to take a pretty large diameter, which therefore is going to overestimate the volume contained within the particle, but underestimate the faceting or surface area. Now, if we then were to put ground coffee on an SEM, we would be able to see that there are regions of really dense, probably small particulates. Then there's this very large piece in the middle that has holes in it. We attribute these holes to where the cells once were. So this is a particle in the vicinity of 500 micron in largest diameter, and the holes are in the vicinity of, let's say, 50 to 100 micron. Okay, but if we zoom in, there's these little pieces of dense material, and these are sort of the fine particulates from the coffee grounds. And according to this paper, the spherical approximation for the small particles holds true for particles that are less than 100 micron. And so then the question is, what are we getting out of our coffee? Well, before I answer that, I also want to put to rest maybe one of your, no, go back 2 slides, excuse me. I want to put to rest one of your thoughts about these pores, right? So these are large pores. And large pores do pose problems for liquid flow through them, but I'm not going to focus on that today. Instead, I also need to make sure that those small pieces are not microporous. In other words, they don't have the same scale of pores as the moth. And the reason for that is we don't want to have diffusion limitations. So we don't want to have molecules that are being extracted from the coffee but then can't make their way out. So we can perform a gas adsorption measurement where here the unit is in milliliters per gram of material, and this is nitrogen. And so we're pumping in, and in this moth, you can pump in about 1 liter per gram, 1 liter of nitrogen per gram of moth. Whereas for something dense like coffee, you can see that you pump in only about 8 milliliters per gram. And so this is actually pretty typical for a dense material. So we're going to make the assumption moving forward that we can approximate coffee as small, dense spheres. To actually make that measurement, I asked Beckman Coulter if they would lend me an instrument. And they did, and they delivered it to a cafe in England. So I did all of the experiments in the foyer of the cafe right here while they were operating. They also gave me a person to operate it, and we ground some coffee and measured what came out. And so the first thing that you might look at is the total counts. This is just the number of particles and their size. And if you look here, you can see that there are very few large particles. In fact, there's about 1 per 100 million small particles. And if you can see they're also centered around, let's say, 20 micron in the vicinity of that. But if you normalize by surface area, those big pieces are really large. And so they contribute between 15 and 25% of the surface area. And then if you normalize by volume, it's not surprising that a large piece occupies a lot of space. So the big pieces end up taking up a large volume percent, and the little pieces don't take up that much. But let's then think about the emergence of this grind profile that I show here in volume percent. Well, of course, the big pieces are determined by the grinder. The finer you grind, the smaller the big pieces get. And so that's what's shown here with 2.5 being a coarse grind, 1 being a fine grind. As I grind finer, the big pieces get smaller. And we can understand that, right? I've drawn a schematic for you here. But what you'll note is the small pieces don't change in size, they just change in population. And the reason for that is that every time I crack the coffee, I get dust forming at that interface. So the more times I have to crack the coffee, the more dust I make, but it doesn't change size. And so interestingly, this makes arriving at an ideal grind setting very difficult, because the big pieces get smaller while the small pieces increase in population. So we have a nonlinear response here. I was also pointed to this paper one time when I showed this to some professors of mathematics at the University of Limerick, who said that this fracturing pattern and this mechanism that I presented aligns very similarly with the fracturing pattern that you observe for volcanic rock when it is ejected out the top of a volcano. Well, there you go. So if you can't control the fine particulates by grinding finer, how do you? Well, you could probably change grinders. I don't have that data in front of me at the moment, but I suspect different grinders and different burr shapes may make different fracturing patterns for the small pieces. But actually, temperature is one really easy way to do it. So if I start out at room temperature, I may get this black curve that it seems to have a hip sort of below 10 micron. And then as I cool down just to freezer, it becomes more skewed Gaussian, sort of a more homogenized skewed Gaussian. Continue to dry ice and then continue to liquid nitrogen. And you can see that this is a very nice skewed Gaussian, okay? So you wouldn't think that that difference makes it's not a big difference, but I assure you it is. You can taste the difference. Now, we can also understand this from some sort of fundamental physical perspective, that at low temperature, you can think of all the atoms in the system vibrating slowly. They don't have much thermokinetic energy. But as we increase the temperature, the crack is going to have to meander, find a different way to get through this material. It may not take the most obvious path. The cracks propagate faster at low temperatures, so it's going to find the edge quicker. And so at high temperature, you tend to see a sort of larger spread of small particulates. And this is true for things like concrete as well. So with that in mind, what you can see here is that helps us understand the hip on this thing. Additionally, if you cool your coffee down from the Arrhenius equation, the coffee is going to last a lot longer, because basically all of the processes that are giving rise to degradation are going to go about half the speed for every 10 degrees you cool it down. And then in addition to that, a lot of the volatile compounds are not going to be volatile. They're going to be condensed. And so if you cool something down from 20 degrees Celsius down to minus 20 degrees Celsius, one could expect a prolonging of the shelf life of coffee seemingly indefinitely. In fact, some cafes and enthusiasts around the world have adopted this. And so I point to a couple. These are friends of mine that have sent me the photos today as I prepared for this talk. And these are various different methods in which they invoke to store the coffee. So you see some examples of centrifuge tubes being used with pre-dosed coffee, some vacuum sealed packs, and even a cafe in Oregon has mounted a mini bar freezer above their grinders so that they can grind directly from the freezer. Okay, I don't have a preference on which one of these you use, but I do want to point to also the fact that this idea of freezing coffee and grinding it while it's frozen has even been incorporated very recently into the design of the cafe, where here the actual counter now includes a freezer where they're pulling coffees out and grinding on demand. And I also want to point to one of my more proud moments, where one of my graphs was actually used in a World Barista Championship routine by Kyle Ramage, who was the US representative in 2017 and ended up coming 6th in the world, and attributed his excellent flavor from his geisha coffee to grinding it frozen and achieving that more homogenized particle size distribution. So congratulations, Kyle, on excellent work. And if you're interested in hearing more about the competition, I'll point you to these two papers here. The science one is a fun little piece about the drink that Jennifer mentioned in the intro. So we now have ground roasted coffee. We need to add water. Now, there are many different ways that one can think about how cations will interact with polar compounds in aqueous environments. I've drawn an extremely simplified version of this. For the chemists in the audience, what I'm invoking here is simply an argument of ionic strength. But for those that are less familiar with this, you can think of minerals like calcium 2+ floating around in water and having some water almost explicitly bound to it. Now, of course, it's transitioning from being bound and unbound constantly. But the important point is I could remove one of these waters and have it bind to something that looks somewhat like water. In this case, an alcohol group, which just happens to be attached to a molecule theaflavin. Now, Diaflavin is one of the compounds that you typically will find in a Yirgacheffe wash process coffee from Ethiopia. And we like it because it tastes like black tea. Well, maybe it doesn't happen as microscopically as I've drawn here, okay? But what certainly happens is that more polar compounds tend to like more polar water. And so as a result, we typically find that coffee molecules that taste good tend to be extracted and taste good in water that contains calcium and magnesium and to some extent sodium. But also at this point, you'd look at something like alpha-pinene, which is one of the operative flavors in chamomile from chamomile tea. Now, this is a hydrocarbon. This thing's not going to be very soluble in water. And if you make the water even more polar, this is going to become even less soluble. And so we reach a disparity in our process for selecting a water for our cafe, because perhaps the water we want to extract coffee may have minerals, but the same water may do a very poor job of extracting flavors from tea. And so there's not exactly one solution. But on the other hand, for every plus, we have a minus, right? We have an anion for every solvated cation. And these are really where we should spend our time thinking about. Basically, the coffee industry is afraid of chloride, Cl-, because chloride is known to catalytically decompose stainless steel. And every single material basically in industrial coffee is made of stainless steel. So you don't want chloride-rich water. So the only other anion really, besides perhaps sulfate, which we'll revisit in a moment, that you might interact with in any situation is bicarbonate. But bicarbonate, you'll remember, is already problematic because this is the example, the textbook example of a buffer. Bicarbonate's job is to try and stabilize pH in an aqueous environment. And the reason it does so is because it's a better acid than water, and it's also a better base than water. So it's always going to be stealing the protons or liberating a proton rather than water. And so if water contains a tremendous amount of bicarbonate, then what you're going to have is relatively low perceived acidity of beverages that you want to taste the acids in. Now, if you remember back to the slide where I showed you what scores high on the score sheet, acid is valued in the coffee industry. So water that contains high bicarbonate is extremely detrimental to tasting those acids. I actually went through and took the reference flavors from all of the outsides of these wheels, and I looked up the natural products that give rise to those flavors. Now, there is a logical gap here that those products may not be the same molecules that give rise to the flavor in the coffee. But that's okay, because this is the best I've got at the moment. So until we can elucidate what gives rise to the peach flavor in a particular coffee, we're going to just have to rely on our pKa values for each of the compounds that are, give rise to the flavors around the wheel. And so with this, I can actually analytically redraw the flavor wheel to change in the presence of bicarbonate. And you can see panels that we might like, for example, citrus fruits and the other fruits like pineapple and pomegranate and cherry. They basically disappear as the carbonate hardness increases. And furthermore, you can even look in the region of nutty, where basically you see things like some of the nut flavors disappear and cloves disappear and so forth. And these are things that we value in the coffee industry. But at the beginning, I told you that the coffees that typically scored the lowest are the ones that have low perceived acidity in the first place, like Brazil, Sumatra, Vietnam, and Hawaii. But now I'll make a case that these are actually the most valuable coffees in the specialty coffee industry, because these coffees never had these panels in the first place, which means that if I were to take one of these coffees, not knowing my water chemistry, and try and brew one, and these coffees may have had cinnamon and chocolate and caramelization notes, there's a good chance I'm going to be able to taste those notes independent of the level of bicarbonate that's in my water. So while we may value these low on the score sheet, they are extremely valuable for establishing specialty coffee as a transferable industry where I can give it to, for example, my parents. I can brew it here in my house. I can give it to a student, and I have no idea what any of you are going to do with it. But I hope that the flavors will at least commute to the way you're going to brew it. But if we did want to be specific, there's a finite amount of acidity in coffee. So if you have too much bicarbonate, you just wipe it all out. So there's going to be almost like a hard stop for the amount of bicarb you want to use. And then at the same time, if you start to make your water too polar, you're going to exclude some flavors in coffee you might like. In addition, you're going to gain access to things that were maybe not intended by the roaster, maybe some residual cellulose type materials that are broken down, who knows. And so as a result, there's going to be an upper limit for the level of divalent metals solvated in the water as well. And somewhere in there is going to be your ideal brew zone. Okay. I want to go back just a moment. I'm sorry. That you'll note that there are a couple of curiosities on this graph. First of all, I present the units of bicarbonate in milligrams per liter, but on the top, you can see that I have bicarbonate as calcium carbonate in milligrams per liter. This posed significant challenges when I first entered the coffee industry, because it turns out that the water filtration industry is built around measuring things as if it were calcium carbonate. And the reason for that is the molecular weight of calcium carbonate is 100. So if you were to boil the water off of, some unknown liquid and there's white residual material remaining, all you have to do is weigh it, and that puts you on this scale almost analytically. But of course, that's problematic because it's very difficult to understand how you could weigh bicarbonate as calcium carbonate. Anyway, the point is that one of the challenges in the coffee industry has been learning to speak many different languages, okay? If you were so inclined, a good experiment that you can run with undergraduates is to teach them serial dilutions to arrive at waters that emulate various different geographical locations. For example, you can take a deionized water and add some minerals to it to achieve something that is representative of Melbourne. And at the same time, you could harden up your water and make something that's somewhat like Los Angeles. And the difference between these two coffees, same coffee, everything, but it's two different brews is remarkable. If you're less interested in doing serial dilutions, you can just simply go and buy bottled water. They'll typically tell you the mineral content. If you want to have an extremely terrible coffee experience, I would strongly encourage you to buy Evian. It has a bicarbonate concentration of 360 milligrams per liter. To flip back to my graph, 360 is so far off to the right of this page that there will be not even a remnants of acid in that coffee. If you want to have a reasonable experience, Dasani, Poland Spring, Crystal Geyser, these are all reasonable waters that you could brew coffee with. And here's an example of a water that would be equally terrible, one that I found while I was in Switzerland. Before I go on to the next slide, I want to point out, by the way, that choosing a coffee that has inherent low acidity, low perceived acidity, like a Brazil, Sumatra, Vietnam, or Hawaii, is a very different thing than choosing a coffee that has high acidity and then turning that acidity off. In other words, tasting its conjugate bases. Conjugate bases taste bitter, they taste chalky. A low acid coffee simply just doesn't have that acidity, right? Well, thinking back to the Romans, they chose to go to this Roman baths because of the water. Well, it turns out that the baths actually have extremely high concentration of sulfate. Now, a chemist, of course, did not write this chart. They're missing the formal charges. But what you can see here is that sulfate is in 891 milligrams per liter. The World Health Organization stipulates no higher than 250 milligrams per liter being safe to consume in small quantities. And the reason is it's widely understood that sulfate has a very large hydration sphere. A lot of water molecules are required to solvate sulfate. And so when you drink sulfate, it will enter your digestive tract, water will flow from in your body to into your intestines, and you will get diarrhea. And perhaps maybe that's why the Romans liked it. I have no idea what their diet was. What I can also point you to on this is that the calcium is in relatively high concentration, but the bicarbonate is unusually low because the anion's made up primarily from sulfate. So this is a really strange water, one that we will almost never see in the United States. And so all you would need to do then is do a bit of reverse osmosis to linearly lower all these relative concentrations down to something reasonable, and you can brew coffee with it. And that's indeed why Kelowna and Smalls, the cafe that I initially began my trajectory in, was making great coffees because they had a pretty nice starting water that was unusual. So we put in a lot of work. So it's finally getting to the brewing. Well, this year we had a paper published in the new Cell Press journal Matter that described the development of a numerical model to understand the extraction of coffee in espresso-type baskets. And a basket is the thing that sits inside this piece of the espresso machine. What you can see here is small pieces and big pieces. And these are parameterized based on the experiment. I can change the size of the small ones, but they don't really change that much unless you include temperature. We change the big ones and then the relative populations. And there's other variables as well, like, for example, Z, which is the depth of the bed. And then there's inlet pressure and outlet pressure. And now if I wanted to convert this model to, for example, a pour over, then I would just change the pressure to make it have zero overpressure from atmosphere, right? So basically, it's a relatively transferable model. But we have to parameterize the output somehow. And so the coffee industry has adopted a method of quantifying how much coffee stuff is dissolved in their water by making a measurement that is basically the same as the Brix measurement used in wine. But the reason it works well in wine is because you start out with, like, let's say 14% sugar, and then after the biology has occurred, you end up with 14% alcohol. So you have a tremendous amount of one molecule turning into another molecule, so you can monitor the change over time. But for coffee, to relate the total dissolved solids, the mass of coffee solvated in water, you have to use some very complicated polynomial, which has been empirically fit. And it depends on both temperature and measured refractive index, which is ND here. Now, based on work from Lockhart at MIT in the 1950s, and several other advances since then, what we basically rely on in the coffee industry is a relationship between extraction yield, which is the mass of coffee that ends up solvated, divided by the mass of coffee that we use to brew it, the coffee in the first place, and then total dissolved solids, which is the mass of coffee that ends up solvated versus the total beverage mass. With these two parameters, basically we can make a measurement of TDS using the refractive index measurement. And that puts us on the y-axis here. On the x-axis, this is a measurement of extraction yield, and this is back calculated. And somewhere in the middle there is thought to be ideal or tasty coffee. So if you were to extract, let's say, 20% of your coffee, and it happened to show up in a concentration between 1.2 to 1.4%, that's probably going to taste good. Well, I'm not 100% sure I agree with that, because the refractive index of pure water is 1.333 at room temperature, but the refractive index is proportional to liquid density. And so if I dissolve ethyl acetate, I'm going to get less dense water than if I were to dissolve sugar or if I were to dissolve sodium chloride. For example, looking at a paper published in 2015, I can dissolve 5 weight percent ethyl acetate and get the same observed change in refractive index as if I were to dissolve 1% by mass sodium chloride. In addition, the refractive index changes vastly with temperature. So what I'm really trying to say is, that using a refractive index measurement to tell you a quality of coffee is basically impossible. And this is really the dagger for that, because as I change temperature, we know from a variety of Nobel Prizes in the 1901 with Van 't Hoff, 1902 and 1904, I believe, showed us the temperature determines the equilibrium constant for a given reaction. And actually, this is really important, because as I change the temperature of an extraction, if I were to take a low temperature brew at 20 degrees Celsius and take that to, let's say, infinity amount of time, what we would get is only about 9 milligrams of caffeine per gram of coffee that we use to brew it in the first place. But if I were to use 90 degrees, I can get something like 15 milligrams of caffeine out of each gram of coffee that I used. Okay, that's fine. There's a temperature dependence. But if I look at a different molecule, caffeic acid, I don't see a temperature dependence. It's within the margin of error for this, for all temperature regimes. So what that means is that changing temperature of extraction changes the ratios relative to one another within the cup. So how is it possible then that one could make a refractive index measurement and make a statement about quality? Well, I don't think it is, but it's what's done in the industry right now. And so as long as it's done in the industry, we're going to try and adhere to that. So for our model parameterization, we only need to fit one diffusivity constant, and we only need to fit one rate constant. In other words, coffee stuff is being extracted. Of course, there's room for improvement, because we want to know what that stuff is. And so I can tell you, I'm really delighted to tell you that as of only a couple of days ago, I've been notified that I've been awarded a grant from the Coffee Science Foundation to go after a real-time measurement to look for qualities in coffee. And with this will, I think, change the landscape of how we discuss and analyze and compare coffees. But with our model, our simplified model, what it tells us is two things, right? We can predict based on a grind size and extraction yield. And so the key take-home message from the graph on the left is that increasing the mass of coffee that we use will decrease the extraction yield. because if we use the same amount of water, we extract less from all the coffee. And at the same time, if we then or let's say, as we increase the pressure, the water flows through the bed faster in our model. And so we end up having less contact time. So we end up extracting less. So our model sounds good. It makes sense physically. And it's this blue line. An experiment basically agrees to a point. And at this point, we see a decrease in extraction yield and an increase in variation. We can attribute this variation to clogging. Basically, let me put it a different way. On this side, an extraction yield that gives us, let's say, 22%, somewhere around here, has some fraction being extracted to 25%, but not the whole bed. And indeed, far more of the bed is being extracted to a much lower percentage, giving rise to an average that is decreasing relative to our model prediction. And so this is pretty interesting because it means that there is a critical point in which you start to become inefficient with your extraction of coffee. In the paper, we go on to suggest two iterative routines to optimize this process. So if all you cared about was extraction yield, which is largely thought to determine flavor, and you might initially like an extraction that tastes good here, one way to achieve the same extraction yield is to grind coarser, find the point in which it starts to tip off the other way. That's where it starts to become more reproducible. And then just simply use a little less water, and that'll drop your extraction yield. Okay, so that gives you a smaller beverage on the top. And if we were to go down the bottom, you could say, okay, well, I could achieve the same extraction yield by grinding coarser and using less coffee, because if I use less coffee, the extraction yields go up, and then I can find out over here, basically use less coffee, grind much, much, much coarser, I can achieve the same extraction yield. But then the concentration of the cup goes down. So interestingly here, what I'm proposing, if you were to iterate on this, this is reducing beverage volume, and this one's reducing coffee mass that you're using to brew it. And so if you were to take this, to continuum, basically what you'd get is the most reproducible espresso is one that involves no liquid and no coffee. And that's obviously a problem, because obviously you want to serve an espresso. And so the companies have to make a decision as to what is a desirable beverage size and a desirable concentration to still have it as an acceptable espresso. Okay, so we're rounding the end of the talk here. So I encourage you, if you have questions, to put them into the chat window. But I want to tell you about something. Some of you may know this, some of you may not, about something very special that I've been able to do because of this unique platform of Zoom. Because we didn't have to fly out, because we've all experienced the shutdown from COVID, some of us have managed to save a little bit of money. We took that money that was going to cost me to go and give the talk in Irvine, and we actually contacted a coffee producer in Colombia to develop us a very special National Academy of Sciences Distinctive Voices coffee, which we have distributed to some of you already. Now, if you missed out on the coffee and you were on the mailing list, I'm sorry, because the type of coffee that we did produce for this is extremely small micro lot, a nano lot, if you like. So there's only a finite amount. And if this is the first you're hearing of this coffee, that's probably because you're not on the NAS's Distinctive Voices mailing list, which is free to join. So I encourage you to not miss out on things like this again and get on that mailing list by going to the Distinctive Voices website. But before I finish up, I want to tell you a little bit about this coffee, because this isn't just me going and buying a coffee. What we did was we leveraged all of the wonderful relationships that we've forged over the years to bring something that is truly once in a lifetime. The competition coffee that Kyle Ramage competed with and the other competitions I've been involved with are basically the same style of coffee as what we produce today. So I didn't produce it, of course. Felipe Trujillo over in Colombia has produced a wonderful geisha. Then that coffee has been imported by Joel Eastlick at Yellow Rooster Coffee Importers. And then that was roasted very graciously by Andrew Sinclair at Mad Lab Coffee in California. And then he was the one who distributed it to you. So along this supply chain, we've had to tap into every single step along the way to achieve excellence. And I want to share with you the processing method. So this is not like the simple one I showed you before. Instead, what we've got now is a geisha that has been handpicked. It has then been both aerobically and anaerobically fermented. And then it has been washed. And then it's been mechanically dried, both air and mechanically dried. This coffee is supposed to be truly remarkable, but I have not yet tasted it because it has just arrived to some of us on our doorstep. And I want to share with you some comments about this processing method from the producer, Felipe, who said that he's looking for exotic flavors like passion fruit and petroleum that comes from the seed. But he chose to do the aerobic fermentation for 3 days to achieve a strong fragrance that is curious, that makes you think, that makes you question. Then he did an anaerobic fermentation for 8 days, which gives you fruity complexity, and he detects notes of yellow fruits and cognac. And then finally, he depulps it, trying to balance the fruity taste with the floral smell. And then in addition to that, not losing the beautiful elegance of a geisha variety of coffee. So with that, I'm so proud to have been able to do this, because this honestly it is extremely difficult to organize something like this, and it was not possible without those 3 extremely important people along the way. So I sincerely hope that if you did receive this coffee, that you will enjoy that. If you want to have a brew recommendation, I'll help you with that. You can email me, or we can talk in the questions. And I want to leave you with a couple of closing remarks. That if you care about water at all, the main thing to concern yourself with is bicarbonate. Basically anything above 80 milligrams per liter and you're in trouble. Secondly, if you've never thought about it before, you should probably weigh the mass of coffee you're using to brew and also the mass of water, so that you can then isolate variables of temperature, time, etc. And finally, I strongly encourage you to always obtain freshly roasted coffee, ideally from local roasters. And I want you to be mindful of the price that you're paying for the coffee, because these coffees, at some point, money has to make it back to the producer, right? So the more money you're spending, in principle, should mean the coffee was worth more to the producer, but you should ask those questions. And finally, I hopefully throughout this talk did not try and tell you that you did anything wrong. In fact, I'm trying to tell you you're doing everything right, because if you like what you're tasting, then continue to do that, because as long as you're tasting your coffee and it tastes good, then that's excellent. So with that, I want to thank a few people. I've worked with several people over the years, the baristas, roasters, academics, and equipment manufacturers and support are listed here. But I want to point out 3 very important people, which was Andrew Sinclair at Mad Lab Coffee in Los Angeles, who roasted this coffee especially for us here at Distinctive Voices. He also roasts other coffees, so please go check that out. Felipe Trillo, who is a producer in Colombia, who I sincerely hope gets to see this presentation. And we've made a point of sending him some of his own coffee, which producers very rarely get to enjoy. So I sincerely hope that he tries that. And thank you to Joel at Yellow Rooster, who without him, we wouldn't have this coffee in America. And then finally, 3 other people who are quite important here: Jennifer, who's joined us again, Tom, and Joseph here. So thank you all. Thank you for your patience throughout the talk, and I'll happily take any questions.